Hello everyone. In this class, we are going to discuss about the structure of proteins. And as we discussed in the previous classes, the proteins are important class of biological macromolecules and made up of are nothing but the polymers of amino acids. And here the structurally the proteins it can be described in terms of four levels of organization, which includes primary structure, secondary structure, tertiary structure, and quaternary structure. And here, as we discussed earlierly, the primary structure is nothing but the having a series of amino acids which are linked one another with the help of the peptide bond and going to make the polypeptide chain, which is called as the primary structure. And the secondary structure means it is a localized spatial arrangement of the polypeptide chain with the help of the hydrogen bonding. And here the polypeptide chain it is locally arranged or undergo spatial arrangement with the help of the majorly with the help of the hydrogen bond and going to form the secondary structure, which includes the alpha helix, which is a helical structure, and the sheet like appearance in the beta pleated sheet. And here these are the example for the secondary structure. And tertiary structure includes the it is nothing but the three-dimensional folding of the entire polypeptide chain. It is called as the tertiary structure. It is going to be stabilized with the help of the many interactions such as the hydrogen bonding, disulfide bonds, hydrophobic interaction, and the salt bridges. So here, with the help of all those interactions, the entire polypeptide chain it is undergoes the folding. So here, the overall three-dimensional folding of the entire polypeptide chain it is going to comprise this tertiary structure, which is stabilized by the with the help of the so many different kind of interaction. Whereas the quaternary structure which includes so here it is restricted to the protein which is and the are made up of the two or three or more polypeptide chain where two or three polypeptide chain are going to interact one another with the help of the non-covalent interaction and going to comprise or make the quaternary structures. And here now we are going to discuss about the secondary structure. So here it is refers to any chosen segment of a polypeptide
It's
chain
15
and
hours.
describes the local spatial arrangement of its main chain atoms without regard to its to the positioning of the side chain or its relationship to the other segment. So here which means the here the polypeptide chain it undergoes the local spatial arrangement with the help of the main chain atoms. So here this spatial arrangement or the localized spatial arrangement it is occurs because of the main chain atoms without regard to the positioning of the side chain or to the relationship ship to the other segments. And here the regular secondary structure occurs when the dihedral angles phi and the psi are remains the same throughout the length of the segment. So here one of the characteristic feature of the secondary structure means so here their dihedral angles phi and the psi are remains the same or the nearly same throughout the segment. So here these are the two angles which is present in the peptide bond, and here the phi is the angle between the hydrogen atom and the alpha carbon atom, whereas the psi is the angle between the alpha carbon atom and the carbon atom of the carbonyl group. And here these are the bonds. So here they can able to undergo the rotation in the peptide bond, and here these due to the rotation of these two angles, so here the secondary structure it is going to form. So here the regular secondary structure there they will be having their characteristic phi and the psi angles, and they will be remains the same throughout the segment. At the same time, the prominent secondary structures which includes alpha helix, beta conformation, and the one more type which is called as the beta turn. Now we are going to discuss about the one of the secondary structure which is called as the alpha helix. Alpha helix majorly it is a left-handed helical structure or the spiral structure, where in this helical structure the side chain of the amino acid residues are going to protrude outside. And here the alpha helix is stabilized by the formation of the hydrogen bonding between the peptide carbonyl oxygen and the amide hydrogen. And usually the hydrogen bond it is going to forms between the peptide carbonyl oxygen of the nth residue to the amide hydrogen atom of the n plus fourth residue. So here between the first residue and the fourth residue, usually the hydrogen bond it is going to forms between the peptide carbonyl and the amide hydrogen. So here majorly the alpha helix is stabilized with the help of the hydrogen bonding. At the same time, the single turn of the helix is extends about 5.4 angstrom along the long axis. At the same time, the each turn which is having the three around 3.6 amino acids. At the same time, as I told you earlier, the secondary structure they will be having the characteristic set of dihedral angles that it is going to define the each type of the secondary structure. And here in this case, the alpha helix also having the characteristic set of the dihedral angles, which includes the phi is having around minus 57 degree, whereas the psi is having minus 47 degree. And here the left-handed alpha helixes are theoretically less stable and have not been observed in proteins. And here this is the structure of the alpha helix. So here it is nothing but the helical segment of the polypeptide chain. So here it is just nothing but the helical segment of the polypeptide chain. As you can see here, these are the R group which is indicated in the green color. So here they are protruding outside. So here the side chain of the amino acid residues are protruding outside. At the same time, we can also able to see that the formation of the hydrogen bond. So here these green dots indicate the hydrogen bond between the carbonyl oxygen and between the amide hydrogen. And here this alpha helical segment is stabilized with the help of these hydrogen bonding between the carbonyl oxygen and the amide hydrogen. So here hydrogen bond it is going to form between the nth residue to the n plus fourth residue. So here usually between the first to the fourth residue usually the hydrogen bond is going to be formed. So here the green color dots which is going to indicate the formation of the hydrogen bond. So here at the same time so here the entire the so here this is the entire turn of the alpha helix. So here which is having around 3.6 amino acid residue, and here the length of this turn is around 5.4 angstrom. Okay, so here how we can able to distinguish between the right-handed helix and the left-handed helix. So here if you are going to hold your hand yeah, yeah, as it is indicated over here, both the left hand and also the right hand. And here if you are going to fold the other four fingers, and here if you are going to facing the thumb away from you. And here for example if you are going to hold the left hand, and here if you are going to fold the other four fingers, and here if you are going to imagine the helical segment or the helical structure which is spiraling around your thumb in the direction the other four fingers are curled or the other four fingers are folded. So here that is going to indicate the left hand helix. Similarly, if you are going to hold the right hand and folding the other four fingers, and here facing the thumb away from you, and here if you are going to imagine the helical structure spiraling around the our thumb in the direction the other fingers are folded. So here that is going to make the right-handed helix. So here this we can able to distinguish between the left-handed helix and the right-handed helix. And here so here after this one, the some of the amino acid sequences are going to affect the stability of the alpha helix. So here the presence of the different kind of amino acid sequence it is majorly affecting the structure of the alpha helix. So for example, a polypeptide chain is there. If it is having the long block of glutamic acid residue, means it is not going to make the alpha helix, because the glutamic acid residue it is going to carrying the negative charge in its side chain. So here these negatively charged carboxyl group of adjacent glutamic acid residue are going to repel each other, and here due to which this polypeptide chain it is not going to form the alpha helix at pH 7. For the same reason, if the polypeptide chain is having many adjacent lysine argine residue, means so here that polypeptide chain also not going to form the alpha helix, because these positively charged residues are going to face the repulsion between them, and here due to which this polypeptide chain is also not able to form the alpha helix. And here at the same time the bulk and shape of the amino acid residues such as asparagine, serine, threonine and cysteine residues also destabilize the alpha helix, because if they are placed very much closer to one another in the polypeptide chain, means so here these amino acid residues also able to destabilize the alpha helix. And here at the same time so here the alpha helix is the helical structure. So here it is undergo twisting. So here the twisting it is also ensures that So here the interaction also going to happen between the side chain of the amino acids. So here, since the alpha helices undergo twisting or the helical arrangement, so here we can also able to see the interaction between the side chain of the amino acid residues, and especially the critical interaction occur between amino acid side chain of the first residue to the amino acid side chain of the third residue or the fourth residue. And here, due to which what kind of interaction is going to happen, and here the positively charged amino acid residues are often found three residue away from the negatively charged amino acid residues, and here by doing so it is going to invert in the formation of the ion pair. And here how the interaction is going to occur means, so here usually the positively charged amino acid residues are often found three residues away from the negatively charged amino acid residues, and here by doing so it can able to form the ionic interaction, and here it is also going to stabilize the alpha helix. And here one more kind of side chain interaction is, so here the two aromatic amino acid residues are oftenly similarly spaced, and here it is going to result in the hydrophobic interaction. So here the spacing of the presence of the aromatic amino acids is going to happen at the similar spaces in the alpha helical segments, and here this is going to ensure the formation of the hydrophobic interactions. And here, apart from that one, certain amino acids are there. For example, the proline or the glycine. So here, these are the two residues are having the least proclivity to form the alpha helices, because in proline, the nitrogen atom, it is a part of the rigid ring, and here the rotation about the nitrogen and also the alpha carbon bond is not possible, because this is the phi bond, which is the characteristic set of the all the different kind of secondary structure. But here this bond it cannot able to rotate freely. So that here thus the proline residue it is going to introduce the destabilizing kink in the alpha helix. So here, in addition to that one, the nitrogen atom in the proline residue, it is not having the substitute hydrogen atom. So that it can able to form the hydrogen bond with the other residue, because the alpha helix. So here it is stabilized. It is because of the hydrogen bond between the carbonyl oxygen and also the hydrogen of the amide group. But here the proline residue, it is not having the any hydrogen in its nitrogen atom. So that here it cannot able to form the hydrogen bond with other residue. So here, for all these reasons, the proline is rarely found in alpha helix. And here, apart from this one, the glycine is infrequently occurs in the alpha helix for a different reason, because the glycine is having the more conformational flexibility because it is a smaller amino acid. And here the polymers of glycine tend to take up the coiled structure. It is different from that of the alpha helix. So here, for this reason, these two residues, the proline and the glycine, they will be having the least possibility to be present in the alpha helix. And so here, one more aspect about the alpha helix that we need to understand is the helix dipole. So here, in the earlier class, we were discussing regarding the peptide bond. So here, the peptide bond, it is having the partial positive and the partial negative character. Because so here the carbonyl oxygen of the peptide bond, which is going to carry the partial negative charge, whereas the nitrogen atom of the amide group of the peptide bond, which is going to form the partial positive charge. And here, because of this one, the peptide bond, which is going to act as the dipole or the electric dipole. And here the electric dipoles of peptide bond are aligned with the help of through the hydrogen bonds of the helix. So here in the alpha helix, as we discussed earlier, the hydrogen bond is formed. So here the electric dipole character of the peptide bond is aligned through the hydrogen bonds of the helix, resulting in the net dipole along the helical axis that increases helix length. So here, due to which what will happens means, so here throughout the length of the helix, so here it is going to create the net dipole. And here due to because so here the partial positive and the negative charge of helix dipole resides in the peptide amino and also the carbonyl groups near the amino terminus and the carboxyl terminus end respectively. So here because because of which, so here the partial positive charge will be created at the amino terminus end, and the partial negative charge will be created at the carboxyl terminus end of the alpha helix. And hence, so here usually due to because of this region, usually the negatively charged amino acids are present towards the amino terminus segment of the alpha helix. So here which is going to create the stabilizing interaction with the positive charge of the helix dipole, and it is also true for the carboxyl terminal end of the helical segment. So here the stability of the alpha helix, it is going to be stabilized, or the five types of the constants, it is going to affect the stability of the alpha helix. So here the one type is the intrinsic propensity of the amino acid residue to form an alpha helix. So here some of the amino acids will not be having any propensity to form the alpha helix. So here apart from that one, the interaction between the side chain or the R groups, particularly those spaced three residues are apart. As we discussed, the hydrophobic interactions are the ionic interactions. So here the interaction between the side chains also in some way it is going to stabilize in the alpha helix, and also the bulkiness of the adjacent R groups also going to affect the alpha helix. At the same time, the occurrence of the proline or the glycine residue is also going to destabilize the alpha helix. And here, apart from that one, the interaction between the amino acid residue at the ends of the helical segment and the electric dipole inherent to the alpha helix. So here, all these five types of the constants are going to affect the stability of the alpha helix.